Good day. My name is Ocean Margate, and together with my group mates Christa Javier and Montebon, we will be reporting about Experiment 12, Redox Reactions and Qualitative Analysis. In this oral report, we will be discussing the introduction of the study, including the principles, significance, and objectives. We will also be discussing the methodology, the results, and discussion for the five tests that were done in this experiment. And lastly, we will be discussing the conclusion and the recommendations. The experiment is based on the concept of redox reactions. A redox reaction is a type of chemical reaction that involves the transfer of electrons between two species. There are two parts for every redox reaction: oxidation and reduction. The oxidizing agent accepts electrons from the reducing agent, which is oxidized. At the same time, the reducing agent donates electrons to the oxidizing agent, which is reduced. Redox reactions play a part in vital, everyday processes such as photosynthesis, respiration, combustion, and corrosion or rusting, to name a few. Thus, understanding redox reactions is essential for understanding the world around us. Redox reactions are commonly applied in the laboratory. This experiment focuses on how redox reactions are used in qualitative analysis and exhibits the role of redox reactions in the characterization of compounds and detection of functional groups.
For the objectives, the experiment aims to differentiate functional groups which can undergo different types of oxidation reactions. It also aims to identify the oxidants responsible for each reaction. Lastly, the experiment aims to compare results of substances with different functional groups.
For the methodology, in this experiment, five tests with different reagents were done to the following samples: acetylene, benzene, styrene, hexane, acetone, formaldehyde, ethanol, ethyl acetate, glucose, fructose, and sucrose. Before any tests were done, the colors of the samples and their reagents were noted. Next one, the sample was collected in a 5 ml test tube, and for samples that were in gaseous form, a stopper was added tightly. 5 drops of an aqueous solution of potassium permanganate was then carefully added, and the changes in the solution was observed and noted. And this process was repeated using the different samples. For test 2, the sample was collected in a 5 ml test tube, and for samples that were in gaseous form, a stopper was added tightly. A few drops of 2 molar sulfuric acid was slowly added into the test tube and was shaken slowly before adding aqueous potassium dichromate. The test tube was then shook for 1 minute, and the changes in the solution were observed and noted. This test was then repeated using the different samples. For the third test, 20 drops of Welling's reagent as well as 5 drops of the sample was added in a 5 ml test tube. This test tube was then heated in a water bath, and the changes were then observed and noted. This was repeated using the different samples. The fourth test was done using Benedict's reagent. In its 5 ml test tube, 20 drops of Benedict's reagent was added together with 5 drops of the sample. This was then heated in a water bath, and the changes in the solution was observed. This was then repeated using the different samples. For the fifth and last test, 20 drops of Toland's reagent was added to a 5 ml test tube, and 5 drops of the sample was then added to that same test tube. This solution was then heated using a water bath, and the changes were observed. This was then repeated using the different samples. Moving on to the results. For the first test using potassium permanganate reagent, the observed reactions were either a purple solution or a colorless solution with brown precipitate. The purple coloration is attributed to the presence of permanganate ions, and the brown precipitate indicates formation of manganese dioxide. The positive result for this test is the presence of brown precipitates, as seen in acetylene, styrene, formaldehyde, ethanol, glucose, and fructose. The result is also the observance of color change. Either a yellow, orange, or a green solution was observed. A positive result for this test is characterized by a green solution as observed with formaldehyde, ethanol, glucose, and fructose. The green coloration is expected when samples are oxidizable primary and secondary alcohols. For test 3, either a blue solution or a solution with reddish-brown precipitate was observed. The blue coloration is due to the presence of the bis tartrate complex of copper and is indicative of a no reaction, while the reddish-brown precipitate is due to the presence of copper 1 oxides, a product of the reaction of the reagent with non-aromatic aldehydes and monosaccharides, as observed with formaldehyde, glucose, and fructose. The observed changes for test 4 was either a blue solution or a solution with brick red precipitate. Same with test 3, the blue solution is indicative of a no reaction, while the red precipitate is due to the presence of copper 1 oxides. This positive result can be observed with formaldehyde, glucose, and fructose. The last test, the observed change was either a colorless solution or the formation of a silver mirror. The colorless solution indicates a no reaction or absence of an aldehyde functional group, while the formation of a silver mirror, as observed with formaldehyde, glucose, and fructose, indicates reaction of the reagent with an aldehyde. Moving forward to the discussion. Bayer's test for unsaturated hydrocarbons is used to determine the presence of carbon-carbon double and triple bonds, given that these bonds are not within an aromatic ring. It can also be used to determine the presence of functional groups such as aldehydes and some alcohols which can be oxidized. The test utilizes a dilute potassium permanganate solution to oxidize a double or triple bond. Oxidation occurs since the carbon's charge becomes +2 instead of +1 as it loses an electron. A double bond will be replaced by a diol to produce an alkene diol, while a triple bond is oxidized to have two hydroxy groups on each carbon or a carboxylic acid. The permanganate ion contributes a deep purple color, and the reduction and oxidation of the hydrocarbons destroys the permanganate and produces a brown precipitate due to the formation of manganese dioxide. The disappearance of the characteristic color of permanganate and the formation of the brown precipitate or color indicates a positive result, while a deep purple color with no precipitate indicates a negative result. Some alcohols, such as ethanol, produce a positive result due to potassium permanganate being a very strong oxidizing agent.
For the second test, we have sulfuric acid and potassium dichromate, which is used to test for primary and secondary alcohols. This redox reaction converts primary and secondary hydroxyl groups into oxo groups. This means that primary alcohols are oxidized to aldehydes, whereas secondary alcohols are oxidized into ketones. A key step in this reaction is the extraction of hydrogen atoms by an oxygen atom to release water. This is extracted from the OH group and the carbon attached to the OH group. For this reason, tertiary alcohols, which do not have any hydrogen atoms attached to the carbon, cannot be oxidized. In the case of oxidation of alcohols into aldehydes or ketones, chromium ions are formed as a side product. This serves as the indicator for a positive result of the test, as chromium ions exhibit a green precipitate. The third test uses Felling's reagent to determine aldehydes from ketones. An exception to this, however, are alpha hydroxy ketones, which react positively to the test, for reasons we will be discussing later on. This test is used also as a general test for monosaccharides. It contains two solutions, feelings A and feelings B, which forms a complex that acts as the oxidizing agent. What occurs in this test is the oxidation of aldehydes to carboxylic acids. Ketones, however, will not oxidize and thus would show a negative result. A byproduct formed in this reaction is copper oxide. Copper oxide exhibits a red color and thus precipitates after the reaction. This serves as an indicator for a positive test result. Fourth test is the Benedict's test. It uses Benedict's reagent to distinguish aldehydes from ketones. Like Felling's test, alpha hydroxy ketones are an exception. This test has a similar use, similar mechanism of action, and a similar reagent with the Felling's test. A key difference, however, is the improved stability of the reagent. In the Felling's test, the reagent requires two solutions to form the oxidizing agent. This is due to the instability of Felling's reagent. However, in Benedict's test, the reagent is highly stable and thus requires less preparation. Similarly, Benedict's test shows the oxidation of aldehydes to carboxylic acid. Copper oxide is also formed as a byproduct, which can be seen as a red precipitate indicating a positive result in test. The fifth test is the Tollens test, which uses Tollens reagent to differentiate aldehydes from ketones. Again, alpha hydroxyketones are an exemption and show positive results to the test. This time, however, instead of copper, silver is used as the oxidizing agent. In this reaction, silver ions are reduced to metallic silver. When presented against glass, this metallic silver forms a mirror-like substance. This is the reason why this test is also known as the silver mirror test. As an oxidizing agent, Tollens reagent oxidizes aldehydes to carboxylic acid. As previously mentioned, metallic silver is produced, forming a silver mirror-like substance in the test tube. Let us discuss the three different tests used to differentiate aldehydes from ketones. In the experiment, Felling's reagent, Benedict's reagent, and Tollens reagent were used to identify aldehydes from ketones. Common to all three of them is the exception of alpha hydroxyketones from the test. The reason for this is the ability of alpha hydroxyketones, or at least some of them, to tautomerize. This tautomerization forms an aldehyde, which shows a positive result to the test. Here are the reagents used in the Felling's test and the Benedict's test. It can be seen that the central atom is copper, which is the reason why the byproduct that allows identification of the positive result is copper oxide for both tests. Some aldehydes, however, resist oxidation by Felling's reagent and Benedict's reagent. An example are aromatic aldehydes. These aldehydes are able to resist oxidation due to the relatively higher stability of the entire compound. The resonance and the structural effects of the aromatic ring prevents weak oxidizing agents such as Felling's reagent and Benedict's reagent from oxidizing aromatic aldehydes. Tollens reagent, however, as a strong oxidizing agent, is able to oxidize such types of aldehydes. A special group of organic compounds are known as carbohydrates or sugars. These are composed of monosaccharides as the most basic units. Examples of such are glucose and fructose. These compounds are composed of a carbon chain, alcohol substituents, and carbonyl groups, existing specifically as aldehydes or ketones most of the time. Monosaccharides containing aldehydes are called aldoses, whereas monosaccharides containing ketones are called ketoses. As basic units, monosaccharides can be linked together to form disaccharides. An example of such are sucrose and lactose. In disaccharides, the effects allowing oxidation of the aldehyde or ketone groups is impeded. This is why, in the experiment, sucrose showed negative results for the tests. Glucose, however, and fructose showed positive results. Glucose, a monosaccharide, contains an aldehyde, thus being classified as an aldose. This aldehyde group is ready for oxidation, thus showing positive results for the tests. Fructose, another monosaccharide, contains a ketone and thus being classified as a ketose. However, it tested positive for the tests for aldehydes. The reason for this is that fructose is a type of an alpha hydroxy ketone, thus showing positive results for the tests for aldehydes. Now, for the conclusion and recommendation, among the functional groups tested in the experiment, alkenes, alcohols, aldehydes, and ketones were discovered to be readily oxidized. Of course, the oxidation of such groups relies on oxidizing agents or oxidants. In the experiment, potassium permanganate, potassium dichromate, Felling's reagent, Benedict's reagent, and Tollens reagent were used to introduce an oxidizing agent. These oxidants vary in oxidizing strength, and this results in different degrees in oxidation. The recommendations, tests for the confirmation of the presence of the oxidized state of the functional groups can be done to ensure that the reaction has taken place. Tests to determine the presence of alcohols, aldehydes, ketones, esters, or carboxylic acids are an example. The effect of the presence of other functional groups on oxidation can also be tested. A good example are aromatic rings, which, as discussed, prevents oxidations of aldehydes from being done by weaker oxidizing agents. Other redox reactions and tests can also be done, an example being the iodoform test. That is all for our presentation. Thank you, and stay safe.